It's my great pleasure to welcome you all for this special session on gene editing, which has been designed to inspire the young minds and to provide a closer look at one of the most transformation technologies of and scientific advancement of our times. This initiative have been aimed to bridge the gap between the cutting-edge research and the school level science education, thereby helping you all to understand how gene editing technologies are shaping the future of health and agriculture and biotechnology as specific. We are truly honored to have with us exceptional panel of speakers, leaders, pioneers who have worked significantly towards the advancement of in the field of genetics and genomic engineering. Today we have Dr. K.C. Bansal as a keynote speaker, former director of ICAR, National Bureau of Plant Genetic Resource, New Delhi. Professor Bansal is a globally recognized expert in functional genomics, genetic engineering, and genome editing for building. I request all the students to please settle down fast, and the teachers to please take a note of the students who are just roaming around, please. Yeah. So PhD gold medalist from IIT Delhi and a postdoctoral researcher from Harvard University, Cambridge. Dr. Bansal is also worked as a visiting scientist at Rutgers University, USA. Dr. Bansal has held prestigious positions including Norman Borlaug Chair and elected Secretary of National Academy of Agricultural Sciences. Currently, he is adjunct professor at Guru Jambheswar University and serves as a board of directors of Global Plant Council and board of trustees of MS Swaminathan Research Foundation. With over 150 high impact publications in journals such as Nature Biotechnology, Nature Genetics, Nature Plants, and PNS, he has played a visionary role in the India's agricultural biotech landscape. With these words, I would request Dr. Bansal to please give a keynote address to the gathering, please. I request all the students to kindly listen. He has much to speak. No? Slides. Oh, okay. Point.
Well, good afternoon to all of you. Sabhi ko mera namaskar. And first of all, I would like to actually, thank the organizers, the Secretary to Government of India, Ministry of Earth Sciences, Dr. Ravi Chandran, my friends in DBT, DST, and all over those who have organized it. And मैं आप लोगों से रिक्वेस्ट करूंगा जो स्टूडेंट्स हैं अगर वो एक नई टेक्नोलॉजी के बारे में थोड़ा जानने की कोशिश करेंगे तो हमें अच्छा लगेगा हमें जिस काम के लिए यहाँ बुलाया गया है हम कोशिश करेंगे कि आपको उस टेक्नोलॉजी के बारे में मैं बताऊं सो बहुत से स्टूडेंट्स हैं जो बहुत इंटरेस्ट लेते हैं अपने साइंस सब्जेक्ट्स में स्कूल्स में कॉलेजेस में अगर कॉलेज स्टूडेंट्स भी अगर यहाँ हैं और हम भी उन्हीं की तरह से उतनी ही इंपॉर्टेंस देते हैं किसी भी नई नॉलेज को हासिल करने के लिए सो टुडे this is a technology which the whole world is talking about it in fact which has been developed only for the last about more than a decade 12 साल पहले ही ये टेक्नोलॉजी बनी है तैयार हुई है जिसको नोबेल प्राइज मिला है दुनिया में 2 women साइंटिस्ट को तो उस टेक्नोलॉजी के बारे में बातचीत करने के लिए आज हम लोग यहाँ एकत्रित हुए हैं एंड आई एम वेरी हैप्पी we have my 2 very good friends as also subsequent speakers, Dr. Ramesh Sonti, who will be also talking to you about this technology further. और मैं आप लोगों को जो आज बताना चाहता हूं मेरी प्रेजेंटेशन में कि हम लोग आत्मनिर्भर भारत तभी बन सकते हैं जब हम अगर अपनी एग्रीकल्चर प्रोडक्शन को बढ़ाएं और इतनी बढ़ाएं कि हमें बाहर भी हम उसको एक्सपोर्ट कर सकें और हम अपनी ही नेशनल नीड्स को पूरी साथ में करते रहें और जो चैलेंजेस हैं उसको कैसे मीट करें और जीनोम एडिटिंग का उसमें क्या रोल हो सकता है। सो मैं आप लोगों को एक ओवरव्यू, एक पिक्चर शो करूंगा, दिखाऊंगा कि what all government of India is engaged with and what are the messages from our top level honorable prime minister to down to the level of our science and technology minister. So if I may start with my first slide, जिसमें हम बात कर रहे हैं आत्मनिर्भरता की बात अगर करें, जो कि इसका जो मिशन है समृद्धि फॉर आत्मनिर्भर भारत, विज्ञान से समृद्धि। सो विज्ञान से समृद्धि को ही ध्यान में रखते हुए हम लोग बात कर रहे हैं कि साइंस हाउ इट कैन मीट द सोसाइटल नीड्स। और हमारी सोसाइटल नीड्स क्या हैं? फॉर एग्जांपल, वी वुड लाइक टू ऑलवेज हैव फूड अवेलेबिलिटी, इस एक्सेसिबिलिटी एंड अफोर्डेबिलिटी, कि हमें वो फूड जिस पर्टिकुलर एक प्राइस पर मिलना चाहिए, जिस क्वालिटी का सेफ्टी फूड वो हमें मिले। देन ऑल्सो न्यूट्रिशनली रिच डाइट हमें मिले, फॉलोड बाय द हेल्थ ऑफ लाइफ स्टॉक and human health itself, which is also again all dependent on agriculture and the food, kind of food we produce. And finally, of course, while agriculture disturbs the environment, but we would like to do that kind of an agriculture, still increase our production so that we keep our environment clean and green. So these are all very important aspects we would like to deal with through science to meet our societal needs, which is also the theme of this particular conference we are meeting here today, of this particular festival, which I am very proud to be part of it. Now, when we talk of the availability of food, India was fortunate to witness green revolution. अगर स्कूल में बच्चों को पढ़ाया गया होगा, मुझे जरूर मालूम है ग्रीन रिवोल्यूशन के बारे में, कि दो साइंटिस्ट डॉक्टर नॉर्मन बोरलॉग और डॉक्टर एम एस स्वामीनाथन, डॉक्टर एम एस स्वामीनाथन जो इंडिया में जो इंडियन एग्रीकल्चर रिसर्च इंस्टीट्यूट है, जो इंडियन काउंसिल ऑफ एग्रीकल्चर रिसर्च है, जो मिनिस्ट्री ऑफ एग्रीकल्चर है, उन्होंने बहुत सालों तक उसको दिशा दी और ग्रीन रिवोल्यूशन को आगे बढ़ाया। और ग्रीन रिवोल्यूशन के चलते हम लोगों की जो प्रोडक्टिविटी थी, खास करके व्हीट और राइस की, वो जो कि एक आधा टन होती थी करीब करीब 1950 में, 3 टन पर हेक्टेयर से भी ज्यादा हो गई। और उसकी वजह से आप देखेंगे कि हमारे यहाँ कभी एक सिचुएशन थी शिप टू माउथ, कि हम लोग अमेरिका से गेहूं आती थी, उसका इंतजार करते थे 60s में, और तब हमें वो कहीं मार्केट में गेहूं जाके मिलती थी। So there was a situation, we used to call it as ship to mouth, because we were not self-sufficient in our own production of whether wheat or rice, which are the main staple crops. But because of green evolution, our productivity went so up from 0.5 ton per hectare in 50s to more than now 3.5 to 4, 5, 6 tons for some of the varieties in our own country, that we are almost sufficient with regard to food production, particularly wheat and rice. And India could enact a law, that right to food act, to everybody has a right to food, that is in 2013. And then, you can see here, without increase in land, हमारी land हमने एक्सपेंशन नहीं किया, प्रोडक्टिविटी इतनी बढ़ी कि हमारी प्रोडक्शन आज अगर आप देखें 2020-24-25 में हम लोग 350 मिलियन टन के पार पहुंच गए हैं, जो कि कभी 1950 में 50 मिलियन टन होती थी। सो आज हम 350 मिलियन टन भी पार कर चुके हैं। और you can also see our honorable prime minister gives a message that to 80 crore of people every month, every day there is a free food available as a result of green revolution and because of the new varieties our scientists keep developing. But then हमें हमेशा हमारा फूड जो है वो बढ़ाना ही है, बिकॉज जब तक फूड अवेलेबिलिटी नहीं होगी किसी भी कंट्री में, एग्रीकल्चर अगर आपका समृद्ध नहीं होगा, तो हम लोग बाकी एक्शंस पर काम या हमारा मन नहीं लगा सकते। वैसे स्पेस टेक्नोलॉजी हो, मेडिकल फील्ड हो, सर्विसेज हो, मैन्युफैक्चरिंग हो, वो तभी सब हम हासिल कर सकते हैं when we have our agriculture making a lot of good progress, we have enough food available, we remain self-sufficient, not only for wheat and rice, for all other major crops which we require in our country, which I'll be talking to you. So here हमारा अभी उद्देश्य क्या है भारतवर्ष का, कि हम लोग हमारी फूड प्रोडक्शन को बढ़ाएं to up to 520 million ton. Currently we are at 350 million ton. Also fruit production भी डबल करना होगा, vegetable production, nutri cereals, millets, milk availability, इन सब की मात्रा 2047 तक जो मैंने फिगर्स दी हैं हमें उनको बढ़ाना होगा. That's something very important to become आत्मनिर्भर. और लेकिन हमारे पास बहुत से challenges हैं unfortunately. You know, challenges हैं कुछ because of the population, because we are the largest population in the world. I'm sure students को ये सब मालूम होगा कि आज के दिन पूरे वर्ल्ड में अगर सबसे ज्यादा जनसंख्या अगर किसी देश में है तो वो भारत में है. India today has even surpassed China, is one of the most populated countries, but at the same time we feel proud of ourselves that we are sufficient enough in food production, at least of wheat and rice. Other crops, there are issues which I'm going to talk to you about. For example, हमारे यहाँ climate change की problem है, natural resources की कमी होती जा रही है, arable land बहुत ज़्यादा हम expand नहीं कर सकते हैं, water की problem है, और आप देखेंगे कि बहुत सी pulses और oil seeds जो हमारी main food है for vegetarian diet, you know, वो हम लोग import करते हैं. और देन हमारे यहाँ एक वर्ल्ड में यूनाइटेड नेशंस ने एक सस्टेनेबल डेवलपमेंटल गोल का एक सॉर्ट ऑफ स्लोगन दिया है बाय 2030 कि हम लोग जीरो हंगर होना चाहिए. किसी कोई भी somebody should not actually speaking die out of starvation. So there should be no zero hunger by 2030, जो कि बहुत दूर नहीं है. So जब इन चीजों की अगर हम बात करें तो और ये से अगर हम हासिल कर पाएंगे तभी we can try to become a self-reliant nation. We can move towards that once we can achieve and can address all those challenges. So then I just want to give you a little bit of information about particularly the import of oil seeds and pulses. हम लोग in general देख रहे हैं आप यहाँ क्या क्या import कर रहे हैं. हम लोग सिर्फ बहुत सी चीजें import कर रहे हैं, लेकिन 60% जो import खर्चा है हमारा, जो foreign exchange का खर्चा है, 60% of it that goes to vegetable edible oils और pulses. Edible oil हम लोग इतना import करते हैं कि 1,57,000 करोड़ 1,57,000 crore every year, about 19 to 20 billion US dollars हम इसमें खर्च करते हैं हर साल. So can we try to increase our own production that we become self-sufficient like wheat and rice, even in our oil seed also? Is there a possibility? So that's what we are genome editing or new technologies can really play a big role. और इस ग्राफ में आप देखेंगे कि हमारी प्रोडक्शन एडिबल ऑयल्स की करीब 10-12 मिलियन टन है, हमारी कंजम्पशन करीब 25 मिलियन टन है. तो जो बीच का जो गैप है अबाउट 14-15 मिलियन टन वो हम लोग इंपोर्ट करते हैं और उसकी कॉस्ट करीब करीब 1.5 लाख करोड़. अगर एक नया एयरपोर्ट हम हिंदुस्तान में बनाते हैं जो कि भी बन रहे हैं इधर उधर बहुत से हमारे अच्छे, उसमें 30 से 40 हजार करोड़ रुपया लगता है. और हम यहाँ हर साल 5 एयरपोर्ट का पैसा सिर्फ एडिबल ऑयल इंपोर्ट करने के लिए लगा रहे हैं. तो हमें इस पर हमें चिंता होनी चाहिए. और इसी के लिए एक्चुअली गवर्नमेंट ऑफ इंडिया ने नीति आयोग में एक रिपोर्ट तैयार किया है कि कैसे हम इस इंपोर्ट से बच सकते हैं, अपनी प्रोडक्शन को कैसे बढ़ा सकते हैं. So here some of the ideas given are listed here. You have to use modern biotechnological tools and you have to improve the production of sunflower, castor, rapeseed, mustard and all other oilseed crops so that we come out of this problem of importing again and again. And therefore, our Honorable Prime Minister has given this kind of a message to all of us that reduce import dependence on agricultural commodities. This is हाल ही में उन्होंने एक ये ऐसा message दिया है. And what he wants us to achieve self-sufficiency in pulses, similar like we have in rice and wheat, so that India must aim to reduce imports and increase our exports. Not only we reduce imports, we increase our exports. हम इतना produce करें, and for which we are capable. We have to develop new varieties by using modern tools of biotechnology. And next is, now we want to aim at increasing our production by 40% in next 5 years. For example, today we are producing only about 24 million ton, and we want to increase it to 35 million tons. This I'm talking here for pulses. This is for pulses. The previous message was for the oil seeds, but pulses also we import, and we heavily import and quite a bit on it. And we would like to expand either the cultivation area or enhance the productivity. But I would like as a scientist to give more emphasis on enhancing productivity, because अगर आप productivity pulses की पूरे world में compare करें, India की productivity is the lowest, less than 1 ton per hectare, whereas बाकी countries में in the world it is even up to 5 to 6 tons per hectare. So there's a lot of potential. So we need to apply science. And similarly, just like pulses, oil seeds में, pulses में भी नीति आयोग ने अभी 2025 में हॉल में एक ये रिपोर्ट तैयार की है. I talked about the pathways for accelerating growth in pulses And again, they're talking about the use of application of modern biotechnology, develop some of those genotypes in new varieties, which are high yielding, which are climate resilient, which is very important. So the point I'm trying to give you all friends, students, अगर आप ध्यान दे रहे हो, तो please ध्यान दीजिए कि what is that new technology of modern biotechnology based technology, जिसकी वजह से हम लोग आज हिंदुस्तान में जो काम शुरू किया है उसके बारे में मैं आपको बताऊं और उसी को अगर हम आगे बढ़ाएं उस काम को टेक्नोलॉजी को यूज करें ऑलरेडी कन्वेंशनल प्लान बिडिंग जैसे मैंने आप लोगों को एग्जांपल दिया ग्रीन रिवोल्यूशन का ग्रीन रिवोल्यूशन कन्वेंशनल प्लान बिडिंग के द्वारा आई थी conventional plan bidding still we continue using it effectively in all the crops but nevertheless we need to accelerate our genetic improvement जो production का rate है उसको बढ़ाने के लिए हमें हमारी जो genetic improvement है उस चीज को बढ़ाना होगा उसके लिए conventional bidding alone may not be enough so we would like to supplement it with for example I have given here genetic engineering or genome editing I am going to talk to you about more genome editing but point is also this कि हम लोग इसके कुछ फायदे हैं genetic gene editing के that this technology is quite precise, fast, accurate. You know what? Students को मैं ये बताना चाहता हूँ कि हम लोग जैसे दिखते हैं कोई भी production किसी भी variety की कम या ज़्यादा tall या dwarf is all governed by genes. I'm sure this you all know. Even a common man must try to know that what we carry and what we look like is all because of genes we get from our parents. So similarly in plants or in any other organism, genes govern any kind of an attribute or property, that organism carries. And if you want to change it, improve it, then we have to do the changes at the level of DNA, at the level of genes. So this technology allows you precisely to do those changes which are required to improve any organism for that matter at the level of genes. पहले ग्रीन रेवोल्यूशन में जब हम लोगों ने वैरायटीज जो साइंटिस्ट मैंने बताया डॉक्टर नॉर्मन बोरलॉग डॉक्टर स्वामीनाथन ने जो बनाई उनको हमें उस टाइम जीन्स मालूम नहीं थे 1960s में हमें था कि इस प्लांट को ये टॉल है इसको ड्वार्फ करना है तो हम फिनोटाइप देखकर बोलते थे कि हमें जो चाहिए वो मिल गया लेकिन आज हमें मालूम है कि कौन सा जीन को अगर हम मॉडिफाई करें तो हमारा प्लांट जो टॉल है वो ड्वार्फ हो जाएगा तो इसलिए जीन एडिटिंग अलाउज यू to make those precise changes accurately in a given single gene, there are about 20 to 25,000 genes typically in any organism. तो उसमें से अगर single एक gene को उसमें भी एक जो हमें exact change करना है to improve a property, we can do it through genome editing. And these are the two scientists, lady scientists who got a Nobel Prize in 2020. The technology was mature enough actually to talk about in 2012. और इस टेक्नोलॉजी से कैंसर भी ठीक हो सकता है इस टेक्नोलॉजी से जो इनहेरिटेड डिजीजेज हैं वो ठीक हो सकती हैं एग्जांपल्स हैं मैं आपको एक दो दूंगा और क्रॉप्स में भी बहुत से इसमें पॉसिबिलिटीज हैं कि हम क्रॉप्स को क्लाइमेट रेजिलिएंट या पेस्ट रेजिस्टेंट बना सकते हैं वी कैन डेवलप क्रॉप्स व्हिच रिक्वायर fewer of fertilizers or pesticides we can ensure higher yields therefore we can save land and also we can use water or any other resources more efficiently एंड दिस इज द टेक्नोलॉजी मैं इसमें आपको डिटेल्स में नहीं जाना चाहता हूं ये टेक्नोलॉजी क्या शुरू होती है कि हम एक डबल स्ट्रैंडेड डीएनए को जो जीन जिसको हम इंप्रूव या उसका कंट्रोल करना चाहते हैं उसका एक्सप्रेशन या उसकी जो प्रॉपर्टी जो फिनोटाइप है तो हमें एक डबल स्ट्रैंड ब्रेक उसमें करना होता है और वो डबल स्ट्रैंड ब्रेक करने के बाद वो एक रिपेयरिंग मैकेनिज्म है सेल में जो होता है या तो आप जीन को डिलीट करना चाहते हैं उस फंक्शन को या उसको आप फर्दर एडिट करना चाहते हैं वो दूसरे ये डिलीट करना चाहते हैं SDN1 ये SDN2 या फिर वो अगर नया जीन जैसे हम GM क्रॉप्स में डालते हैं नया जीन भी आप डाल सकते हैं सो दैट बिकम्स अ GMO लेकिन हम लोगों ने हमारे यहाँ कोशिश की कि इंडिया में जबकि GM क्रॉप्स या वर्ल्ड में कहीं भी GM क्रॉप्स रेगुलेट होती हैं जिसमें 10-12 साल लगते हैं वो एक एसेंशियल प्रोसीजर है टू टेस्ट द बायोसेफ्टी ऑफ दोज न्यूली डेवलप्ड GM प्रोडक्ट्स whether it is biosafe to human beings, to animal, to livestock, and to the environment, or to the soil microbes, or to many other organisms. So they all undergo regulation, whereas, these SDN1 and SDN2, through genome editing, we thought as scientists it should not be regulated because they are non-GMOs. But in 2020, in our country, we did not have those guidelines well defined about genome editing. Those guidelines were developed. And we thought, we had a discussion among scientists in the National Academy of Agricultural Sciences that why should we regulate SDN1 and SDN2 when we do not have a foreign gene present, in these final products. Okay, and we only make small changes within the native genes of the organism. So finally, we had a discussion, in our National Academy. I was personally, out of my own interest, actually involved in it and sending a message across, even requesting Government of India to exempt, SDN1 and SDN2 from biosafety point of view. And finally, in fact, the landmark decision came, on March 30th, 2022, that government agreed to our discussion on scientific basis, and they exempted, SDN1 and SDN2 की उसकी कोई बायोसेफ्टी रेगुलेशन की जरूरत नहीं है, क्योंकि हम कोई जीन बाहर से नहीं लेकर आ रहे हैं. और फिर बाद में हमने एक रोड मैप बनाया कि इंडिया में एग्रीकल्चर में किस तरह की जीनोमेरिटिंग किस क्रॉप में होनी चाहिए. खास करके हमने ध्यान इसलिए दिया, ऑयल सीड्स पर आप देख रहे हैं सबसे प्रायोरिटी रखा है ऑयल सीड्स में, मस्टर्ड, सोयाबीन, ग्राउंडनट, क्योंकि वो इंपोर्ट करते हैं. नेक्स्ट पल्सिस पर जोर दिया है, चिकपी, पिजन पी, ग्रीन ग्रैम और भी हैं. और देन ऑफ कोर्स राइस, व्हीट हम इग्नोर नहीं कर सकते हैं क्योंकि हमारा स्टेपल फूड है. सो फिर हमने एक प्रोजेक्ट बनाया इन 5 क्रॉप्स को लेकर, राइस को साथ लिया. ऑफ कोर्स राइस में ऑलरेडी एक बहुत अच्छी प्रोग्रेस हो रही थी. आईसीजीबी में इंस्टीट्यूशन जहां से डॉक्टर रमेश सोंटी हैं और हमारे आईसीआर सिस्टम में भी. तो राइस के साथ हमने फिर मस्टर्ड, सोयाबीन, चिकपी और पिजन पी पर भी लिया. और इसको एक बहुत अच्छा ये प्रोजेक्ट बना 2022 में, जबकि एग्जाम्पशन हमें मिल चुकी थी उस टाइम. ये अगस्त में बनाया, अगस्त 2025 जो कि हो चुका है, ये अभी दिसंबर तक भी चल रहा है ये प्रोजेक्ट. और इससे 2 वैरायटी वर्ल्ड में फर्स्ट टाइम जीनोम-एडिटेड वैरायटीज को इंडिया ने लॉन्च किया. ये वैरायटीज के फायदे हैं, इसमें आपको यहाँ दिखाया हुआ है, that they can produce more, about 90 to 20% higher yield. They are tolerant to salinity and alkalinity, and they mature 20 days earlier. One of the varieties, which is, this one, DRR Rice 100, it matures about 20 days earlier, so saves a lot of fertilizers, lot of water, which is important, and it gives you finally a good income to the farmers. There is a possibility, of course, it has to still go to the farmer's field, but there is a strong possibility that the farmer's input will be reduced and output will be more, and they get more yield and more benefit from economic point of view as well, more profit. But then more rice varieties are also being developed in several institutions. They are listed here, like IRI में, IIRR, which is in Hyderabad, Central Rice Research Institute in Cuttack, Tamil Nadu Agriculture University in Coimbatore, and ICGB, जहाँ से डॉक्टर रमेश सोंटी यहाँ के डायरेक्टर हैं, वो नेक्स्ट टॉक देंगे. वो भी एक्टिव फील्ड ट्रायल में थर्ड प्रोडक्ट, यू कैन इमेजिन, आफ्टर दीज टू दैट विल बी द थर्ड प्रोडक्ट ऑफ राइस व्हिच इज undergoing field trials. So which is a very good progress, I would say, with regard to our moving ahead. But important is our focus, we should always remain more on oil seeds and pulses. So oil seeds में जो एक प्रोडक्ट 2 राइस के बाद अगर आ सकता है, तो वो जिसमें डॉक्टर नवीन बिष्ट, I must like to congratulate him, who is present here, who is a moderator for the panel discussion. His lab, he has developed this first, product. This one he has developed, जिसमें glucosinolate actually is an issue, whether in the oil quality or in the oil meal quality. But उन्होंने एक नोवेल्टी से काम ये किया है कि उसकी जो ग्लूकोसाइनोलेट की जो क्वांटिटी है उसको लीफ में डिस्टर्ब नहीं किया, लेकिन ऑयल सीड में कम कर दिया. हम लोग नहीं चाहते थे कि वो लीफ में कम हो, क्योंकि दैट आल्सो प्रोवाइड्स यू रेजिस्टेंस टू पेस्ट. Enough glucosinolate which is present in leaves, it gives you enough protection against pests. But we don't want that zero, we want almost in fact in seeds. So that's what he has achieved. And this is, after these 2 rice varieties, this is the third product which is going to second year of ECLIPS trials, by ICR. Next is in NIPB as well. Another mustard variety is being developed with higher oil content, as you can see here. And there's a lot of effort on pulses in many of those institutions. You can see on pulses as well. There's a lot of effort going on here. And also, of course, in different institutions, I should mention those institutions. They're all listed here, of course. Also, in Nabi right here in Mohali, and there's more research going on. But important is, meriting for climate smart agriculture. And because you can see here the temperature in the last about only 25 years, how much it is increasing compared to what before the industrial area, industrial revolution, we had this kind of increase in temperature was not there. But now you can see the temperature is increasing to the level of 1 degree, which is very high, actually speaking, and affecting the production of, our crops in different districts. You can see here, and Bio E3 policy has got a climate resilient agriculture as one of the themes. Bio E3 policy is an important policy which was launched by Honorable Prime Minister in August last year, 24. And then we have therefore a big project now, rupees 500 worth crore rupees project for enhancing climate resilience in about 40 crops, 24 field crops and horticultural crops, 16 of them, and so many institutions already working on it. And then now the idea is to give more emphasis on increasing the efficiency of nitrogen, phosphorus, and potassium as shown here. And because we are dependent on import of fertilizers as well. You can see here, nitrogen we are importing so much. In fact, we are importing here so much. Yeah, we are importing here nitrogen. You can see we are importing so much. Even not only nitrogen, not only urea, even muriate of potash and all of the fertilizers we are importing. And one of the scientists from again NIPGR, where once Dr. Sonti himself was directing that research, he was head of the institution. This wonderful piece of work has been done for increasing the phosphorus utilization efficiency. Phosphorus is a big problem. We are importing it, so we would like to have more yield with less phosphorus application. And that's what he has done. You can see here, there's more number of tillers, 26% more grain yield in the kind of edited rice which he has developed. Of course, that is now going to newer varieties. So finally, there's a way out to fix atmospheric nitrogen also by plants like wheat, rice. It has been demonstrated. It's a novel technology done by University of California. One of the scientists actually who happens to be a good friend, Dr. Eduardo Blumwald. So we would like that India also must try to do this kind of research, that not only in pulses, even in rice and wheat, we are able to have this atmospheric nitrogen fix so as to have less application of fertilizers. And that's what actually the work I thought would like to be done. And we would like to also take up this agenda forward we have plenty of genetic variation available in our gene banks. So this is what my last slide would be, I think, that we have those plenty of plant genetic resources. We must try to utilize them and finally develop improved crop varieties for different traits we are trying to look for. Some of those genes, naturally occurring genetic variability, we must try to understand it, we must try to mimic it and translate it to any new product through genome editing, which actually speaking, we should be leaders because we have the maximum amount of crop diversity in a single gene bank in the world. And so finally, some take-home messages for some of those scientists, those who are here, or the students, if they want to join research, we always look for some good students. That first is that we must focus to raise our productivity on oilseeds and pulses. We must work to increase or, fix atmospheric nitrogen in crops like rice and wheat or maize, improve efficiency of nitrogen, phosphorus, potassium, and reduce import dependency, and of course, emphasis on climate resilience and nutritional enhancement. And of course, this is again mostly for the scientists that we must work on a fast track mode development. We not work only in silos, we must work in tandem. All institutions working in silos in a way. I think that's not the proper way. We must have single India team. If such a Nobel Prize winning technology, we want to harness its potential, we must use that technology to become Atmanirbhar, particularly with agricultural production. And some of the scientific bottlenecks, I don't want to go into details of this. And of course, we have one of the scientists from Cuttack, Dr. Kutub Mola, has come up with a new enzyme altogether, nuclease, which is not patented in India itself, which we can utilize. And finally, this is the message from our Honorable Minister, Dr. Jitendra Singh, that India's bioeconomy is going to touch $300 billion by 2030. So it's a big field. Biomanufacturing is coming in a big way. It's a wonderful opportunity to deal with agriculture or healthcare or energy or environment conservation. And when we talk of healthcare, using this technology, genome editing, one lady who was having a problem of sickle cell disease, she has been cured, 50 years old lady, Victoria Gray. She is with the Nobel Prize winner, Dr. Jennifer Doudna. This has happened for the first time. There are many other examples of this kind, but time will not permit me to go into the details. And that's where finally our Honorable Minister also gives a message that we are moving towards an era of personalized medicine. So genome editing, sickle cell disease, wonderful work being done here by Dr. Debojyoti Chakrabarti in IGIP, one of the institutions of Government of India. So there's a lot of potential attached to this technology, particularly in agriculture, on healthcare, or for livestock. And message here is that this technology is going to stay, and we must make maximum use of it to increase our production, particularly I'm talking about agriculture. And I remain personally thankful to Dr. Jennifer Doudna, the Nobel laureate, because I had an opportunity where she invited me to her lab. We interacted with her. She keeps on helping on human resource development in providing some of the reagents which are important, for our Indian researchers and scientists. And with this, I thank you. And what you see here, this red flower is actually these white petals. Through through CRISPR engineering, they have become like rose. This is a wonderful thing I thought I'd just share with you. Finally, those students, we organize capacity building every year, 1 or 2, and I would request all of you, those who are interested, just starting actually tomorrow, online. And I thank you very much for giving this, attention to me, and thank you for your patient listening. Students, शायद आप लोग मेरे साथ होंगे, कोई क्वेश्चन होंगे, हम आपको साथ में जरूर क्वेश्चन को आंसर करना चाहेंगे. क्वेश्चन जरूर पूछना चाहिए. मुझे लगता है जैसे हम लोगों को भूख लगती है, हम खाना खाते हैं, ये भी भूख आप लोगों को लगनी चाहिए कि ये नॉलेज मुझे नहीं है, क्यों नहीं है, मुझे पता होनी चाहिए. तो आप लोगों को क्वेश्चन पूछने का ये इतना बड़ा मौका मिला है, इतनी बड़ी कॉन्फ्रेंस गवर्नमेंट ऑफ इंडिया ऑर्गेनाइज कर रही है एवरी ईयर. So students, teachers, science teachers and all a common man should really take full benefit of it. It's very important. हम लोग अपने भारतवर्ष में अभी तभी आगे बढ़ सकते हैं जब हम इकट्ठे मिलकर पूरा आगे काम, साइंस को लेकर आगे काम करें. Thank you very much. Thank you very much. Thank you again.
Thank you, sir, for that insightful lecture on gene editing on agriculture. Next, we have invited speaker, Dr. Ramesh.
Sir, there are few questions.
Sir, after this. After 2 more speakers, we will have a question answer session. Yeah. So next speaker, invited speaker happens to be Dr. Ramesh Sonti, sir, Director of International Center for Genetic Engineering and Biotechnology. Dr. Sonti holds a PhD in bacterial genetics from University of Utah, USA, and completed postdoctoral research at MIT Cambridge. In 1993, he joined CSIR CCMB Hyderabad and worked as a scientist and group leader. His work has significantly advanced the understanding of plant-microbe interaction and biotechnology-driven crop improvement. He has served as director of National Institute of Plant Genome Research, Delhi, and he has been a professor at IISF Tirupati. He is a recipient of prestigious Santi Swarup Bhatnagar Prize. He has awarded DBT, Biotech Product Process Development and Commercialization Award also, and he has fellowship from all the 3 national science academies. Dr. Sonti continues to contribute to the advancing genetic engineering and crop science in India. I welcome sir for his speak, please.
Good afternoon, students. Do you know what a gene is? How many of you know what a gene is? Raise your hand, please. Students, you all know what a gene is. Tell me, what is gene made of? Is it protein? Is it DNA? Somebody said DNA? It is DNA. So genes are made of DNA, and genome editing is a method of modifying the DNA so that you can have better plants, better microbes for industrial microbiology, and for curing genetic diseases. So the next 10 minutes or so, I will tell you how genome editing works and what are the products that are available currently that are being worked on in India to help genome editing applications, and how we can better Indian society using genome editing. So that is what I'm going to try to tell you about. So first part is, what is genome editing? Dr. Bansal has told you that it's a way of changing DNA. That is exactly what it is. But I would like you to tell you that it is like scissors and a pencil. Now, if you write with a pencil on a piece of paper, can you take a rubber and remove it? Yes or no? Yes. So genome editing is like that. You are using a rubber to remove some alphabets in DNA and put new alphabets in DNA. So you are rewriting DNA. Just as you can write with pencil on a paper and use a rubber to remove it and now write again, same thing you can do with DNA. That is what genome editing is. You are rewriting DNA. You are rubbing DNA, some bases in some alphabets in DNA and adding new alphabets. That is what genome editing can do. And when you change the gene, you change the product. You change the property. So that is what I am telling you here. So this is DNA here. And this is the CRISPR molecule. So this is actually what will come and edit the DNA. It is like scissors. You can see tailors using scissors. Same thing, the genetic engineer will use scissors. And the scissors are the CRISPR molecule. So it is this CRISPR molecule that the scientist will use to change the alphabets in DNA. So where is genome editing being used now? Dr. Bansal has told you extensively about applications in agriculture. But genome editing is used not just in agriculture. It is used in industrial microbiology. It is used to make new products, new materials, new clothes, new plastics. Everything that you make using chemistry, you can now make using biology, using genome editing and synthetic biology. So I will tell you very briefly about applications in agriculture, in industrial biotechnology to make biofuel, and in medicine. Very briefly, I'll touch upon each of them. So first thing about genome editing and agriculture is that you have to remember that the crops made using genome editing are no different from the crops that you made using conventional plant breeding. Conventional plant breeding has been done for 100 years and it is very safe. Genome editing is the same as conventional plant breeding. The product is the same. The process may be different, but what comes out at the end is the same as conventional plant breeding. So essentially, Government of India has said crop plants developed through genome editing are no different from conventionally bred crops. So that is the important take-home message. That is why we don't need to regulate them. They are the same as conventionally bred crops. We have been eating them for 100 years or more. So genome editing is the same. Now just one example in ICGB, Dr. Bansal has talked about several other applications. From the institute that I have come from, Dr. M.K. Reddy is a scientist there. He has actually made a rice line that is high yielding using genome editing. This is the wild type rice variety. You can see the amount of grain here. This particular line, students at the back, is this yielding more? This is wild type, normal crop. This is genome editing crop. Which is yielding more? Wild type or genome edited crop? Tell me. Those who agree with me that it is genome edited crop that is yielding more, please raise your hand. More students, raise your hands, please. Yes, good. So genome editing now actually will help you increase yield. This is just in rice. Dr. Naveen Bist has been doing it in mustard. Other people are doing it in pulses. So all of this work is going on in our country. And after the genome edited crop is developed, then it goes through field trials. The Indian Council of Agricultural Research tests it in the field, make sure it is actually working. Only then will it give it to the farmers. So a lot of testing is done before any crop developed in the lab goes to the field. So that's the first part, application in agriculture. Now I'll tell you two applications that are being done in ICGB on industrial microbiology, how to make biofuels. Now before I get there, this is what I was telling you. The Government of India has said that genome edited plants can be considered to be equivalent to traditionally bred varieties. And there are standard operating procedures have been developed by the government to make sure that genome edited lines are actually equivalent to conventionally bred varieties. So when a genome edited plant comes into the field, rest assured that it is the same as conventional bread varieties and it is safe for you to eat. Now I will tell you about how genome editing, this is about agriculture. Now genome editing is being used to make things that will make other kinds of differences in your life. One of them is called biofuel. Normally when we get petrol, how do we get it? That oil comes from the earth. They are oil rigs, the drilling machines that will bring the oil from deep in the earth outside. So all of the carbon that is inside, when that goes in, the petrol goes into the car, the carbon dioxide comes out and it creates pollution. If you go to Delhi or you see pictures of Delhi, there is a lot of pollution now. How do you reduce pollution? By preventing use of conventional fossil fuels and using what are called biofuels. So in ICGB, we are making a yeast. Now you know the yeast, this is what you use to make your bread, to make cake. It's called baker's yeast. That same yeast, we are actually making it for industrial microbiology. So it won't come into your food chain, it will go to a factory. And in that factory, what it will do is it will make diesel. This fatty acid ethyl ester is diesel. So the diesel is being made by the yeast. It is not coming from deep in the earth. There is a bacterium, there is a yeast that takes sugar from a plant, makes diesel, and that diesel goes to your petrol pump and that it will come to you. So this is something we are working on, and in a few years' time, the diesel that you have, part of it will be coming from yeast. Now this is another important activity. There is a compound called butanol. Yesterday, if some of you were there in the bioeconomy, we discussed about how bioethanol is being made. Bioethanol is now part of your petrol. It is coming from plants. Ethanol is being made, that is added to your petrol. But biobutanol is even better than ethanol. So at ICGB, using CRISPR, 8 different genes from another bacterium have now been introduced into E. coli. And now this E. coli makes butanol, and we want to take that and put it into your car so that you get clean energy. And how are we doing it? We are doing it using CRISPR. And this can help combat global warming. So in a few years' time, again, we hope that this is research being done. In a few years' time, we hope that it will come to the market. The last part I want to talk about is, Dr. Bansal mentioned sickle cell anemia. Now this is a terrible disease. And it comes from your genes. The child that has sickle cell anemia, it's no fault of the child. The child is born with a genetic defect. The gene is there on chromosome 11 and there is a defect there. This is the normal red blood cell. This is the sickle cell anemia red blood cell. And if that mutation is there, sickle cell anemia comes to the child. And that child, when they grow up, or as they grow up, they get pains, they get anemia, and they get more infections. So it's a very, very difficult problem. Government of India has launched what is called the sickle cell anemia mission to reduce the burden of sickle cell anemia in our country. So through that process, in that program, all branches of government are working together to reduce sickle cell anemia. And Dr. Bansal mentioned that in the CSIR, at the Institute of Integrative Genomics and Biology, they have developed a new CRISPR tool. That tool is actually being used. Many arms of government are working, and currently at the All India Institute of Medical Sciences, the trials are being done. The CRISPR tool is being used to this particular mutation that causes sickle cell anemia. That gene is being corrected. So in the child or in the adult who has sickle cell anemia, the CRISPR tool will go and it will correct the defective gene. So now there is a new gene and that patient can live a normal life. That is the promise of genome editing. We can discuss more as we go along. Currently, I'll just end here. Thank you all for your attention.
Thank you, sir, for explaining or helping the students to understand the gene editing in such simple terms and examples of pencil and rubber, which you have explained, and their applications in health, agri, and industrial microbiology. Next, we have Dr. Rahul Purwar as an invited speaker, professor at IIT Bombay and founder and CEO of ImmunoAct. Dr. Purwar is a pioneer in CAR T cell therapy in India. He holds a PhD in molecular medicine from Hanover Medical School, Germany. After completing his postdoctoral fellowship at Harvard Medical School, Boston, he worked as a scientist in Immunogen, USA before joining IIT Bombay. His team has developed India's first endogenous CD19 CAR T cell therapy platform, leading to the creation of ImmunoAct, the country's first cell and gene therapy company. Their CAR T cell therapy received Indian market authorization and was dedicated to the nation by the Honorable President on 4th of April 2024, a landmark milestone in gene therapy landscape in India. Dr. Purwar's research work is well recognized in scientific community with numerous publications to his credit in esteemed journals. His contribution to the field of bioscience and bioengineering are commendable, and his work continues to inspire many in the scientific community, and I'm sure the students will love the talk.
Thank you so much, Dr. Rajesh, DBT, and different Department of Government of India for having me here. And I think this is going to be one of my most difficult talk, basis the audience. I will try my best to speak less of science and try to tell you the story which we have been working on in last 11 years. Especially, I will focus on the journey part, how we have developed India's first gene therapy, what Dr. K.C. Bansal talked about and what Dr. Sonti talked about, how we worked very, very hard at IIT Bombay, Tata Hospital in Mumbai, and developed this gene therapy for curing the cancer. आपने सुना होगा कि कैंसर क्योरेबल नहीं है. कैंसर एक बार किसी को डिसीज का पता चलता है तो थोड़े दिन के बाद आई थिंक सब कुछ खराब सा हो जाता है. बट यहां पे एक होप है कि जीन थेरेपी, क्रिस्पर, लेंटीवायरस और डिफरेंट जीन थेरेपी के प्लेटफॉर्म कैंसर जैसी डिसीज को, सिकल सेल जैसी डिसीज को एटलीस्ट क्योर कर सकते हैं. सारे लोग फिर भी क्योर नहीं होंगे, लेकिन कुछ लोग क्योर हो सकते हैं. That's the journey I will tell you in next 10 to 15 minutes, right? हम सबको पता है कि आई थिंक इंडिया इज फार्मेसी ऑफ द वर्ल्ड. हमने कोविड के टाइम पे कोविड वैक्सीन सारे देशों में भेजा, इंडिया की मैन्युफैक्चर. बट हम जहां पे लैक करते हैं वो जगह है इमर्जिंग थेरेपीज. बहुत अच्छी अच्छी जो टेक्नोलॉजीज हैं जो कैंसर जैसी डिसीज को, सिकल सेल जैसी डिसीज क्योर कर सकते हैं, हमारे देश में नहीं है. यहां पे हम लोगों को कुछ काम करने की जरूरत है. और आईआईटी बॉम्बे, जैसे हमने कहा, टाटा हॉस्पिटल, इम्यूनोएक्ट एक कंपनी स्टार्टअप जो मैंने खुद फाउंड की अपने और लोगों के साथ में, हम लोगों ने कुछ काम किया पिछले 12 साल में, और कैसे हम इस टेक्नोलॉजी को लैब से लेके लैब मार्केट तक लेके गए, राइट. कार टी थेरेपी रियली एक एवोल्यूशनरी प्लेटफॉर्म है. इनफैक्ट साइंस मैगजीन ने से लास्ट 100 इयर्स में मेडिकल इनोवेशन का सबसे बड़ा मेडिकल इनोवेशन इन 100 इयर्स में हुआ है तो एक कार टी सेल है, as per Science magazine. This technology basically developed in University of Pennsylvania in US for the first time. Carl June, who treated first patient in 2005 or so, and then he worked also very hard and take this therapy. Now they are curing thousands and thousands of patients. ये थेरेपी एक पर्सनलाइज्ड मेडिसिन है. जस्ट टू गिव यू एन आइडिया कि पेशेंट हॉस्पिटल में आता है, पेशेंट के ब्लड लिया जाता है, और उस ब्लड में जो टी सेल्स है, इम्यून सेल्स हैं पेशेंट के, उन्हें कैंसर फाइटिंग सेल्स हम बनाते हैं. एक बार हमने कैंसर फाइटिंग सेल्स बनाए लैब के अंदर जीन मॉडिफिकेशन जीन एडिटिंग से, ये सेम पेशेंट के सेल्स उसी पेशेंट में दिए जाते हैं। तो पर्सनलाइज्ड इसलिए कि सेम पेशेंट के सेल्स, unlike your paracetamol or the Gentech or whatever the medicine chemotherapy you take, this medicine is personalized medicine, right? ये लिविंग ड्रग है, तो एक ही बार ट्रीटमेंट लेना है, एक बार सेल्स आपके अंदर गए, वो सेल्स अपने आप प्रोलिफरेट करते हैं, अपने आप रिप्लिकेट करते हैं, तो आपको बार बार ट्रीटमेंट लेने की जरूरत नहीं है। तो ये इसका फायदा है। कार टी सेल थेरेपी एक तरह से एक हेल्थ केयर का रेवोल्यूशन है, पर्सनलाइज मेडिसिन है, और एक कार टी केवल एक दो कैंसर में ही नहीं, अभी हाल में ल्यूकीमिया और लिम्फोमा ब्लड कैंसर में बहुत अच्छा काम करती है, लेकिन दूसरे भी कैंसर में इसका काम चल रहा है। न केवल कैंसर, सिकल सेल डिसीज जैसे जेनेटिक डिसऑर्डर्स, आपका हार्ट अटैक, आप अपने देश में हार्ट अटैक की बड़ी प्रॉब्लम है। हार्ट अटैक कोलेस्ट्रॉल लेवल से ज्यादा होने से होता है, तो कोलेस्ट्रीमिया भी इससे कार टी सेल से ठीक हो सकता है। तो ऐसे कई सारे इनोवेशन लैब में क्लिनिकल ट्रायल में हो रहे हैं, लेकिन आज की डेट में जो मार्केट में अवेलेबल है वो ल्यूकीमिया और लिम्फोमा के अगेंस्ट कार टी अवेलेबल है। ये एक प्रूफ ऑफ कंसेप्ट है जो मैं आपको बता रहे थे, मोस्ट फिनोमिनल एडवांसेस इन मेडिकल साइंस इन 100 इयर्स। प्रूफ ऑफ कॉन्सेप्ट ये है कि जनरली कैंसर को जो डॉक्टर्स यदि यहाँ पे हैं तो वो लोग कहेंगे कैंसर रेमिशन में होता है लेकिन क्योर नहीं होता कभी। तो ये एक पहली बार एक एग्जांपल है कि एक एमिली वाइटहेड, द गर्ल इज एमिली वाइटहेड, शी वाज द फर्स्ट वन, a kid who was treated by CAR T cell, right? So every year she was going to University of Pennsylvania for this photo shoot, and they will write 1 year cancer free, and after 10 years for the first time they call it cured. Now this girl is cured. She was one of the first one treated by this technology called CAR T cell therapy. CAR T बहुत एक्सपेंसिव ट्रीटमेंट है। एक ट्रीटमेंट की कॉस्ट ऑलमोस्ट $500,000 $500,000 का मतलब हो गया अराउंड 3 से 4 करोड़ रुपए अपने देश में। 4 करोड़ रुपीस का एक इंजेक्शन है, क्योंकि एक ही बार लगता है, एक इंजेक्शन है 4 करोड़ का, तो हार्डली हमारे देश में हमारे पेशेंट इसे ले पाएंगे, राइट? सो फार यू सी देयर 7-8 CAR T अवेलेबल इन द वर्ल्ड, all cost $500,000, एंड 4 करोड़ का ट्रीटमेंट केवल एक ड्रग कॉस्ट है, इसके ऊपर क्लिनिकल कॉस्ट भी है, तो पूरा ट्रीटमेंट का कॉस्ट लगभग 7 से 8 करोड़ आता है। 8 करोड़ का ट्रीटमेंट हमारे देश में शायद ही कोई ले पाता, तो हमने 12 साल में इसको 8 करोड़ 5 करोड़ का जो ड्रग कॉस्ट है उसे बहुत ही 25 से 30 लाख रुपए में हम लेके आए। और ये नेचर मेडिसिन ने ये आर्टिकल भी इसके ऊपर लिखा था एक बार, नेचर ने, कटिंग एज CAR T कैंसर थेरेपी नाउ मेड इन इंडिया एट 1/10 ऑफ द कॉस्ट। ये थेरेपी जब हमने 12 साल में बनाई तो एमिली वाइटहेड जैसे ना हमारे पास भी थोड़े से एग्जाम्पल हुए। जस्ट आप थोड़े एग्जाम्पल शेयर करते हैं। ये प्रोफेसर है न्यूजीलैंड से, 11,000 किलोमीटर न्यूजीलैंड से चल के इंडिया आए। इन्होंने CAR T सेल थेरेपी मेड इन इंडिया बेंगलोर में ली, और नाउ ही इज ऑलमोस्ट मोर देन 1 year कैंसर फ्री। नाउ ही गोज बैक टू यूनिवर्सिटी एंड अगेन बिकम द टीचिंग स्टूडेंट्स एंड हेल्पिंग देम। So many example. This lady Henrik D. Reed, she came to India from Netherlands and got the CAR T in Delhi in one of the hospitals, which is made in India CAR T again. So many patients now, besides our own patients, many patients coming from different part of the world to India and getting the CAR T cell therapy. We are hoping this is our first very little kid, little girl who we treated 3 years ago. 3 साल पहले, she was one of the first kid, little girl from Nashik. She got treated by CAR T. She got leukemia. और now 3 साल के बाद ये अपने स्कूल जा रही है। कैंसर नहीं है अभी इनको, We are hoping she will be our Emily Whitehead after 10 years. She grows up, do something good in her life, and become role model to our generation that a cancer survivor from Make in India CAR T cell therapy. So many different examples. The gentleman, you see a lab technician from Madhya Pradesh, from a very not so much wealthy family, but now you see he also got the CAR T during the clinical trial stage and now cancer free. These are not just few examples. Now we have been doing this in the clinical trial as well as last 2 years and the commercial scale. Now we have so many examples, whether it's a national cyclist, one of our cyclists got into this problem of cancer, now he is cancer free, whether dentist back to her clinic after getting the CAR T and now she is cancer free. The kid on the right, you see a young, handsome man from an elderly parents, got into the leukemia again. So, and now happy back thinking about Bollywood now. We have treated poorest of the poor to the wealthiest of this nation. And now those stories you will find on the YouTube and Google. But what I want to tell you, the message, take-home message is that what once used to be science fiction, today it's a reality in our country, that our country, we also can do such complex therapies, not just in US or Europe, like $1 million treatment at fraction of the cost. We have so many patients, so many people, and cancer is on the rise. Many people will have cancer, unfortunately, because infectious disease are going down. People will have cancer because they are in long age. And this problem of even cancer is on the rise. Many people die in our country more than the other countries. And why people in our country die more from the cancer than other countries? I don't want to have you this, that don't worry about the names, but what I want to just highlight, think about this drug, think about one medicine, and the second medicine, this one medicine, the ऊपर जो लिखा है ब्रेंटुक्सीमैब, 2011 में यूएसए में एफडीए ने अप्रूव किया. 2011 में जब धोनी ने वर्ल्ड कप जीता था, जब तक ये इंडिया आया, रोहित ने वर्ल्ड कप लिया था. Those who knows cricket, you know that it took a decade for the therapy to come approved for our patient than the United States. It takes a decade and sometimes more than a decade for the treatment to come to India and make available to us. Now this is not acceptable to India. We are a nation, we are good in science, we can develop our own treatment, and that was the motivation for us to develop this kind of treatment. And that's where these are the different motivations. And like we all know that we can develop something remarkable at fraction of the cost. And this is what, I hope this motivate some of you. And I will not go in detail a lot, but just to tell you that why CAR T-like treatments are not available in our country. The major bottleneck is cost. Pharma companies, the big players, they don't see market in our country, and that's the reason why they don't ever come. Because $1 million treatment, nobody will be able to afford, nobody will make money in it. So if we have to solve our problem, we have to work hard. We can't be rely on the other nations and other people. So Atmanirbhar Bharat, I think Honorable Prime Minister's thought, I think is very well taken. This work, because it was $1 million treatment, we had to develop at fraction of cost in a small time. We had to have multiple institutions in India, within India, outside India. We all have to work together. So here, IIT Bombay, TMH, National Cancer Institute in the USA, and ImmunoAct, which I founded with some of the people. We all work together, academia, industry collaboration, multi-country collaboration. We all worked hard. And then the team and advisors, current and past, many people come on board, including the Carl June, who developed this therapy in USA. They also come as a team and as a collaborator and mentor. You need a village to solve a problem. So take home message for the student is that if you want to achieve something good, find your good mentors. We developed the CAR-T finally, and first CAR-T, the name is NextCar 19. And some of you can, if you get time, look at the Google, check it, what it does, how it works. But this is entirely make in India treatment, and no work happens very quick. Success doesn't come easy. It took us almost more than a decade. 2013, I came back from US, joined IIT Bombay as faculty member. Since then, we have been working very, very hard to develop the prototype, then going to the translational work, going to the investor, being a IIT Bombay faculty member. I have no idea how entrepreneurship works, but finally, I think government is very supportive. IIT system is very supportive. So we finally have a private limited company as well, which took this work, take it to the masses. We treated more than 500 plus patients now across the country. So what used to be called a boutique therapy in the West, in India is no more a boutique therapy, because earlier there was nothing in India, but now you say, make in India, make in Mumbai, therapy goes across the country in east, west, north and south. Everywhere it is now available in different hospitals. It is not only available in tier 1 city, even in your city, we have treated patients in PGI Chandigarh. The therapy really cost very little, even less than 90% or more than 90% cost reduction compared to US. The results are published in Lancet Hematology journal, which is considered a prestigious journal. So Indian science is giving you as good efficacy as the rest of the world, showing that yes, a good science, good standards at the lower cost. I will not bore you again here with these all the evidences, but just to give you an idea that we have treated many patients on this technologies, especially the blood cancer, leukemia and lymphomas. One question which some of the student will feel, what happens when you develop something of that remarkable at lower cost? Are you going to reduce the quality? This I will put a thought for you to think. This is lower cost doesn't mean low quality. And these are the two images which one of my friend took from the Fairfield Inn in Marriott in New York when he was visiting. He's also from Mumbai. He took this photograph in the morning in the breakfast, and then on the right hand side, at the same cost, you can get same Marriott chain hotel called St. Regis in Mumbai, where you can get the same buffet at the similar cost. So in India, you can develop a good product at reasonable cost. We got a lot of chance to meet the many people. If some of the students are interested, we went to Kaun Banega Crorepati also to meet Mr. Bachchan. So you can see those videos if you are interested. We have now we are sending this therapy to Mexico. So Mexico recently almost like approving the clinical trial. So we are going to treat patient in Mexico. Mexico, just for some of you who do not know, is just next to United States. They share the border. So almost 24 hours of the flight, it will go, make in India product will ultimately make to the Mexico. We are already sending the product to many countries in the GCC region, but now this would be the first when we will send product to very far away. So with this, I will stop here, and I would just say innovation with the purpose and hope for all. Thank you.
Thank you, sir, for taking us through the journey of developing the therapy to defeat the cancer. Definitely bringing the cost down from crores to lakhs of rupees. And by the examples which you have provided of the individuals who have been given the treatment and are doing well. And the last but not the least, the make in India for rest of the world is really remarkable for the CAR T-cell therapy. So before we break this invited lectures part, can we have one or two questions, please? Not more than that. If any students want to ask any questions from the speakers, because we had some students. Please.
Good afternoon, sir. Sir, I have a question for you. Like, we are working on blood cancers. CAR T cells is working for blood cancers. But what about other cancers, such as pancreatic cancers? The cancer cells they just protect themselves with other cells. How do CAR T would reach those cancer cells and work there? Is there any
Sure. So the question is that cancer works for the CAR T works for the blood cancer, but it may or may not work on solid cancer. Or what are the available examples of the CAR T which works for the solid cancer? So yes, CAR T currently works for the blood cancer as well as solid cancers. The solid cancer CAR T yet did not reach to the market. Certainly that CAR T works very, very well in the blood cancer. The efficacy rate or the response rate is very, very good. But in solid cancer, the CAR T is not as effective. And the problem is that when people say CAR T doesn't work in solid cancer, the problem is that CAR T works in blood cancer very, very nicely. It's like response rate can go even 70 to 90%. So almost 60 to 70 or 90% in subset of the patient, they can get benefit from the CAR T in blood cancer. In solid cancer, that's not the case. For example, in glioblastoma, which is one of the most difficult brain cancer, which is not treatable by any other disease, any other modalities, CAR T works, but only in 20 to 30% of the patients. So CAR T does work in the solid tumor, but not at the same response rate as the blood cancer. But yes, many trials are on the way, and I am very sure that in some time, some or the other CAR T will reach to the up to the market stage as well.
Thank you, sir.
Good afternoon, everyone. My name is Kavita. I'm from Punjab University. I'm a PhD scholar. So, sir, my first question is that recently India has patented a gene editing a gene editing tool like TNP based tool. Recently, it has been patented from India. So, what are the challenges that exist for that tool when we compare it with CRISPR-Cas that has already been widely used for gene editing overall? So globally, how can we enhance its capacity as compared to the CRISPR?
Yeah, that's a very good question. And in fact, you know what? This TNPB is 1/4 smaller in size, than Cas9 or Cas12 for that matter. So one of the advantages is that it can be therefore delivered to the plant cells or any other organisms also much easily. And you can carry much more cargo along with it. At times, we don't want to only create a cut, which TNPB or Cas9, Cas12 will do. At times, we want to regulate the gene expression as well. So for regulating the gene expression or for base editing for that purpose, we need to have deaminases or some of the transcription factors or repressors, along with this particular kind of enzyme. So that becomes much easier. But only thing is, yes, we cannot say that how more effective or efficient will it be compared to Cas9, Cas12. It needs to be further tested. Lab work is going on. In fact, we are very fortunate, and we must congratulate the scientist, Dr. Kutub Mola, who has really speaking, identified it and, patented it in India. And we are very lucky. We would like to do more work in Indian labs. Is already starting. Only yesterday we had a review meeting, and we are going to provide some kind of an extra funding into the scientists, to these institutions, more who would like to really test its efficacy. And we start using it, effectively in our Indian system for different crops. So that's what, but in the meanwhile, I must tell you, we would continue using Cas9, Cas12 because time is a factor. We want to be really speaking, move ahead compared to many other countries. Though even if it is patented, never mind, we would like to continue using those. But you're right, TNPB or many other such candidate genes, we should discover more. आत्मनिर्भरता की तरफ अगर हमें बढ़ना है, हमारे यहाँ बायोलॉजिकल जो डाइवर्सिटी इतनी ज्यादा हमारे देश में है, उससे भी हमें और भी इफेक्टिवली नए जीन्स निकालने की जरूरत है और नए न्यूक्लियसेस आइडेंटिफाई करने की जरूरत है. And PhD students like you should really take a lesson and a force, full kind of an exercise you must all do together and see the inspiration which I get from Dr. Rahul. As he's mentioned it, really speaking, we should try to make it effectively as a team and move forward. Thank you. And if there is any question, we can discuss further offline.
Sir, this is with respect to जैसे आपने बोला आत्मनिर्भरता की तरफ हमें बढ़ना है, तो लास्ट में हमने देखा कि हम हर एक प्रोडक्ट की कॉस्ट कटिंग करना चाहते हैं, ठीक है, so that it is affordable to the Indian population over here. So if we are from the science fraternity, we know that now most of the journals where we are publishing, they are paid journals now. Everybody is asking for money, and $1,200 to $3,000 is the minimum amount there for a open journal. So what is India doing in this respect also? Starting their own journals so that they are more affordable. Whatever research is not getting published comes into the public domain. So I want your comments on that too, please.
See, there are journals, if you are doing the open access, then your fees is very high. But in fact, the Lancet Hematology, what I have shown, we haven't paid a single penny for the publication. So there are journals where you don't need to even pay a lot of money. There are some where you have to pay a little bit. But if you want an open access journal, there you have to pay a lot of money. So depending which journal you are talking about, the fees will vary accordingly.
Sir, I worked for Pfizer for production of penicillin G, for yeast, for all these products. My question is, India treats a lot of disease with herbs. Do we plan to do some gene editing so that the efficacy of herbs increases and subsequently herbs will be used as a food for treatment for patients like diabetes? My humble question.
Yeah, thank you. I'll try and answer this question. Already genome editing is being used in several medicinal plants. There is a program of the Council of Scientific and Industrial Research. They're working on several plants to do what they call metabolic engineering. Make mutations in particular genes so that there is more of the active product that is being produced. And I think maybe Naveen Bist will also talk about something that he is doing. Several experiments are on, and hopefully the products will come to the market soon.
This work is in progress in CMAP, one of the institutions, and also National Botanical Research Institute in Lucknow. And wonderful piece of work. They have got also a niche program, particularly on genome editing, as Dr. Santi mentioned.
Good afternoon, sir. My question is slightly different from but is related to this session. This is related to the quality of produce nowadays is produced, and the common citizen in the country is now feels that adulterations and the food quality is not up to the mark, and this is a concern of all of us. So where are we heading to this check? Maybe from the government side or because health implications are manifolds, and even if we go to the markets, we see milk everywhere, paneer is everywhere, though we don't have the productions of this dairy products. The production is not that much. So I think this is the main concern we need to address and focus.
ठीक है, ये आपकी बात
I request that since we are running out of time, so
right. Quickly, क्योंकि आपने क्वेश्चन उठाया है जबकि रिलेटेड नहीं है सेशन के, but then adulteration किसी के कंट्रोल में नहीं है, सिर्फ हमारे ही हम लोगों के कंट्रोल में है. By the way, when we talk of Government of India, there is an act, Food Standard Safety Authority Act is there in India. अब वो एक्ट नया है, बहुत पुराना नहीं हुआ है, और अपनी they are all talking about food safety for sure. They are taking very many steps forward. और ये बात आपकी ठीक है कि हमें उस पर ध्यान देना चाहिए, and I'm sure government of India is aware of it. We all scientists are aware of it. I think important is also for consumers to know not to accept anything, from some of those sources which are not really authorized, and we should try to be more knowledgeable and ourselves as civil citizens try to make a
Yeah,
difference. But thank you, we can talk more.
yeah,
Thank you.
actually, we are running out of time,
Sir,
so
one
it's a humble
last
request.
question I want to ask.
Be very quick because
Okay,
we are
yes.
already running of time.
I believe this is a very important question and it should be considered. It question is divided. It has a suggestion as well as a question. Sir, I'm a postdoctoral fellow working in Delhi University with Maharishi Kanath Postdoctoral Fellowship. I'm working on bacteriophages. बेसिकली थिंग इज दैट कि एएमआर अभी आ गया है तो ऑल्टरनेटिव सब लोग ढूंढ रहे हैं अपने अपने तरीके से. वी आर वर्किंग ऑन बैक्टीरियोफेजेस व्हिच आर 100 इयर्स ओल्ड. वायरस बोल के किसी को देंगे ट्रीट करने के लिए तो कोई लेने को तैयार नहीं होता. थैंकफुली दे गो बाय द नेम ऑफ बैक्टीरियोफेजेस, सो पीपल आर लेस स्केप्टिकल. उनको मालूम नहीं है क्या है बिल्कुल ठीक चीज. लाइकवाइज सर, जेनेटिकली मॉडिफाइड क्रॉप्स के लिए देयर इज सो मच स्केप्टिसिज्म. 2002 से DMH11 is on moratorium, BT brinjal is on moratorium. I am a proponent of biotechnology. I have degree in biotechnology. But thing is that हम इतना पैसा, इतना समय उस चीज में इन्वेस्ट करते हैं, and it leads to nowhere. Everything goes down the drain. BT cotton भी known food है. We don't have any GM food crop. जो abiotic stresses हैं, जो food में नहीं जा रहे हैं, उनके लिए तो we can propose the genetically engineered crops.
So you're right. I think because the time is short, what I will do, I will take one part of your later part of your question. You know that there is no problem with regard to biosafety of genetically modified foods at all. If it is there, it is only because of the spread of misinformation throughout in the world. You must please listen to me what I'm saying. Genetically modified food खाना एकदम safe है. 25 साल से ज्यादा अमेरिका में पपाया जो कि कोई उबालता नहीं कच्चा खा रहे हैं. हम भी हमारे यहाँ खा रहे हैं बहुत सी, खास करके जो बीटी कॉटन ऑयल है, तो उसमें कोई बायोसेफ्टी रिलेटेड इश्यू नहीं है. नंबर वन, आई वांट टू डिस्कस मे बी लेटर. और बीटी ब्रिंजल पर मोरेटोरियम है. DMH11 मस्टर्ड पर कोई मोरेटोरियम नहीं है, वो खाली अंडर लीगल रिव्यू है. But then what I'm saying is people have their own right. It's a democratic world we are living in. So we have to answer each and every question. Even the government is also for, social acceptance. We don't want to thrust upon. थोपना नहीं चाहते हैं. लेकिन scientifically, genetically modified crops हो, gene edited technology हो, CAR T-cell therapy ho, they're all actually speaking science making progress. So it takes time for any technology to get accepted. So, but the only part is that like you and me must not spread misinformation. We must say that science is doing its job, and not that you are doing it. I'm just talking in general, and we must not listen to those activists, unfortunately. Their agenda must be something different, but we scientists must try to spread the real scientific information that genetically modified, genetically engineered, genome editing, all products, particularly I'm talking of food safe, is all safe. Thank you. Maybe bacteriophage question if Dr. Santi can.
Yeah, I think bacteriophage therapy is a very old therapy. Actually, it started many years ago, but because of the success of antibiotics, and how well they were actually working for a long time, it went by the way that attention was not paid to it, sufficient attention. But now that we have this problem of AMR, many places in the world, including in India, are actually trying out bacteriophage therapy. And I think in a few years, it will also be a reality.
Yeah, thank you all for being such a great audience. I extend my sincere gratitude to all the invited speakers being on the dais. Now, may I request Dr. Rajesh sir to present memento to our esteemed speakers. First to Dr. Bansal sir. Thank you, sir. To Dr. Sonti, sir. To Dr. Purwal, sir. Thank you all. Now we will be having the quick panel discussion session. So may I request to Dr. Ark Subra Ghosh sir, Dr. Mohan Kumar sir, Dr. Mahendra Sonwane sir, and Dr. Naveen Chand Bish sir on the dais for quick panel discussion.
Request students to kindly be seated. The teachers may please ask the students to. We have another 30 minutes of panel discussion. 30 minutes. And I'm sure that it will be quite informative as the first session it went by. I request Dr. Vivek to kindly welcome our panel members with a bouquet of flowers. Dr. Arka. Dr. Mahendra Sonawani. Dr. Mohan. And Dr. Naveen Bisht. Let me introduce to all the panelists we have today. Dr. Arka Shubra Ghosh is Director and Senior Principal Investigator at GROW Research Laboratory, Narayan Nitrale. He leads cutting-edge programs in gene therapy, ocular biology, and regenerative medicine. His team has built India's first regulatory-approved AV gene therapy vector production platform to produce cGMP-grade vectors and has also developed hybrid dual AAV vectors. He co-authored India's national gene therapy guidelines and is widely recognized for diagnostic and therapeutic innovations in eye diseases, including point-of-care biomarker kits, clinically adopted formulations for dry eye disease and ocular inflammations. With over 160 publications, including in Nature, Nature Medicine, Nature Cell Biology, he is a leading figure in India's gene therapy ecosystem and is actively involved in mentoring graduate students, scholars, clinicians, and postdoctoral fellows. Dr. Mohan is scientist at Center for Stem Cell Research, which is a unit of iBrick in STEM Bangalore. Dr. Murugasen is a distinguished scientist in therapeutic genome editing for hematological disorders. His research focuses on developing genome engineering platform for diseases such as beta thalassemia, sickle cell disease, and hemophilia. As one of the pioneers, investigators advancing clinical grade genome editing approaches in India, his work holds a potential to revolutionize the treatment options of inherited blood disorders. Dr. Mahendra Sonawani is a professor at Tata Institute of Fundamental Research, Mumbai, and is a leading development biologist working on zebrafish to understand how genes regulate epithelial and gut development. Trained in laboratory of Nobel laureate Professor Christian Nuzelian Wohlhalt at Max Planck Institute, he began investigating the genetic regulation of developmental processes using zebrafish as a model organism. Since joining TIFR in 2009, Dr. Sonawane has built a strong research program focused on uncovering the genes and mechanisms that regulate the development and organization of epithelial tissues. His group now explores how nutrition influences gut development and metabolic health, fostering collaborations between clinicians and basic scientists. He is a recipient of Ramanujanam Fellowship and Wellcome Trust DBT India Alliance Senior Fellowship. Dr. Sonawani is also widely recognized for his mentorship and fostering interdisciplinary research. Our next panel member and the moderator of this session is Dr. Naveen Chandra Bist. He is a senior scientist at National Institute of Plant Genome Research, Delhi. He is a plant geneticist working on plant secondary metabolism and nutritional genomics in Indian oil seed mustard, as the speakers in the earlier session did mention. He has been pioneering, contributing to understand the molecular regulation of glucosinolate diversity in Brassica species. Using CRISPR-Cas, his lab has developed transgene-free low glucosinate mustard lines, which are undergoing ICR field trials. In his recent work, he has demonstrated biofortification of Indian mustard with a cancer-preventive glucosinolate glucoraphanin by precisely editing the AOP2 gene family. Dr. Bist's scientific exhibition is reflected in his high-impact publications, patents, and prestigious awards, including DBT Tata Innovation Fellowship, DBT S. Ramachandra National Bioscience Award, DBT IYBA, NASI, Scopus Young Scientist, and INSA Young Scientist Award. He has been also elected fellow for all the leading Indian science academies. I request Dr. Bist to kindly proceed.
Thank you, Dr. Rajesh. And it's a nice platform to talk about gene editing, and I'm very much thankful to all the speakers who speak about gene editing in crops as well as very recently CAR-T therapy work. So today, although we have very limited audience now, but I'm sure these speakers, I welcome them to have any questions, whatever is there in their mind, because we want more interaction in this session, at least for next half an hour, what will go. But to set up the stage, so we start with CAR-T therapy, what has been doing by Dr. Rahul, and I invite Dr. Arka to speak something more about his approach of doing some of his recent research, which has been highlighted in many platforms, and what he's trying to do with gene editing. So, Arka, can you say something more on what kind of different human disorders you are trying to kind of work on using your approach? Yeah.
Right. So, in our laboratory, we work on eye diseases as well as muscle diseases, brain diseases, and blood diseases. So, in retinal dystrophy, this is a group of diseases जिसमें लोग अंधे हो जाते हैं, या तो पैदाइशी अंधे होते हैं या तो अंधे हो जाते हैं over time. So it is a genetic disorder in all or most of these cases. Or so you can use gene editing tools or gene augmentation tools to correct that. Apart from that, we also are trying to treat sickle cell anemia with gene editing in collaboration with Devajyoti's lab. But we are trying to do in vivo gene therapy, where patient will come, they'll get a single injection and they'll go. So the treatment will happen inside the patient's own body rather than ex vivo gene therapy. And then we work on gene editing for brain disorders. So that's one thing. And the biggest area of work currently is muscular dystrophy. So Duchenne muscular dystrophy, which affects boys only. And it affects, generally the life of a DMD patient is between 20 to 30 years. So that is a huge population in India that has that. So we are trying to come up with the first gene therapy clinical trials for them.
Yeah, so good to listen about the gene therapy trials about muscular dystrophy. So I remember Devajyoti's lecture, and he was also trying to kind of present ये जो case study है of a girl who was kind of suffering from muscular dystrophy. आप थोड़ा audience को उसके बारे में और थोड़ा detail में बताएंगे how you came to this patient, and then what was the approach, and by the time you were getting approval, what all difficulties and challenges you were facing during that particular case study?
Right. So that particular case, it's a case of a child who had a mutation in this gene called SERPINI1. So this is a mutation in the mutation causes the problem in the brain, so in your brain cortex. तो ये family दिल्ली की family है who approached Debojyoti, and through Debojyoti approached me as well to try and create a therapy for this child. So the child was normal till I think 7th or 8th grade, and then she had started having problems in school, and then her grades started going down, and she used to have these seizure-like episodes. So it was treated as seizures or something of that sort. But it took almost 3-4 years before they were able to diagnose in New York that this was an ultra-rare condition called FENID. It's a familial encephalopathy. So essentially, there was a problem in the brain cortex. This protein SERPINA1 was aggregating, और उससे फिर मतलब जैसे उसका her eye was perfectly okay, मतलब उसको दिखाई देता है, but brain is not able to recognize the image, so therefore she couldn't see. Her ear was okay, but she couldn't hear. तो by the time हम लोगों से मुलाकात हुई उसकी, she was already had the cognitive ability of a very small child. Okay, वो खुद से कुछ नहीं कर पाती थी. So it was a race against time. So Debojyoti created these base editors, so an adenine base editor, but my job was to create a drug out of it so that it could actually treat the brain in that patient correctly. So वो हमने within a year's time we did all the developmental work. वो mouse models में test किया कि कौन सा route of delivery, what dosage, does it work in the brain cortex or not. We tested those, and patient derived iPSC cells, उसमें से neurons बना के उसमें disease model किया, because it's an ultra rare disease, तो animal model नहीं था. तो करके हम पहुंचे उस स्टेज तक कि हम सिडेस्को में रेगुलेटरी अप्रूवल के लिए अप्लाई किए, along with a clinical team which was going to do the surgery to do the provide the gene editing therapy in the brain. But at that point, unfortunately, the patient passed away because of aspirational pneumonia, so we couldn't actually give it to her.
Yeah. So yeah, this was something very interesting story, which which we came to know through many presentation of Dr. Debojyoti. और मैं audience को बताना चाहूँगा, we have kind of collaboration with Dr. Debojyoti also. So Dr. Debojyoti has recently not recently, almost about few years back, he has kind of discovered a Cas protein which is from a bacterium, and he has patented it. So जो हमको issues हैं in crop में कि हम कोई भी CAS system को use कर रहे हैं, it is from Doudna's lab or some other place like Broad Institute. So there will be we do not have freedom to operate for commercialization. We are working with Dr. Debojyoti, and we have some interesting result on that particular CAS system which Debojyoti has tested. And Debojyoti का जो system है that Arka is also using, and he is using his delivery method. That is some smartness. Their collaboration is all about that. Debojyoti's reagents he is trying to deliver in site-specific manner कि वो जिस particular organ में चाहिए वो जाएगा, and then the cure of that disorder or disease can be done. Very interesting, Arka. Very, very nice to listen from you. Dr. Mahinda, so I was following your work on zebrafish, and then, yeah, it's quite interesting model. But for audience, we would like to know what exactly you are trying to do in the developmental side of zebrafish. I'm sure development is very important for everyone, be it plants, be it animals, be it zebrafish or any system. And your system using genome editing and all other different approaches are very kind of appreciable. Can you please explain
Thank
more?
you, Dr. Naveen. So as many of us know that the life begins with fertilization. So you have egg and sperm. And the fertilized egg will eventually form an embryo, which will eventually grow into adulthood, and then forms our body. So when this one-celled zygote, which transforms into an embryo, it has to now give rise to multiple different tissues, including your gut, your heart, lens, brain, skin, all these things. And how these things form is basically what developmental biologists study. And we are looking at gut and epidermis, how these two tissues form, and what are the genes which are important for formation or development of these tissues and organs. and how they become functional, because for them to become functional, they have to acquire specific forms, right? I mean, gut is a tube, how you acquire this gut uh, tube. So these are the kind of questions that we ask. And so to understand the genes or the signaling pathways, जो कि इसमें इन्वॉल्व होते हैं, उसके लिए हम CRISPR-Cas9 जैसे मेथड्स का यूज करते हैं. CRISPR-Cas9 से you can inactivate the gene function and ask कि उसमें अभी क्या डिफेक्ट आएंगे. अगर मैंने एक जीन डिफेक्टिव कर लिया है, जैसे कि मायोसिन जीन है या कोई भी जीन है, देन वो डिफेक्टिव जीन से होके उसका क्या असर हो सकता है गट पे, उसके फंक्शन पे, ये सब चीज़ों के बारे में हम अभी जानने की कोशिश कर रहे हैं. Quite a bit is already known. We are adding to that kind of knowledge. But another aspect that is important, so why zebrafish? So zebrafish is important as a model in this. Now, you can't do these experiments in humans, so you have to have animal models. And zebrafish is particularly useful for genome editing or gene editing using CRISPR-Cas9, other editing technologies that exist. And the simple reason behind it is that you can inject the ingredients, so to say, which are important for genome editing, like your Cas9 protein or your guide RNA, that they can be injected directly in the embryos or in the eggs. And so these can function and give rise to loss of function phenotypes, as we call them. But that's one thing. Another thing which is important to note here is that the development doesn't stop when the organism just grow, is born, or, when the growth happens, the development continues. Even in us, some of the tissues are still remodeling, like your gut is still remodeling, and depending on what you eat, it might take different form. In the sense, it can change its architecture. So we are also now looking at in those aspects.
Yeah. So good to know about zebrafish model. Besides zebrafish, are you also trying to use some other non-model organisms specifically, or cell line, where you try to kind of look for these developmental defects? Is it so?
No, we haven't started looking in the. So we have started now getting into organoids.
Okay.
So the idea is to take human cells, especially enteric cells, and see whether we can grow them in petri dishes. Because since we work on gut development, which is not easy to kind of investigate in humans, and if you have human patients with some disease, gut diseases, those cells can be taken, cultured, and can eventually be tried what's going wrong in those. And with even with, one can build the models where once you have the organoid, one can inactivate a gene function and try to understand, or even make a disease model, for example. So there is a disease called as microvillus inclusion disease. It's an enteric disease. And the babies born with these diseases typically die due to dehydration and, also because of malabsorption. And there is no remedy as such for this disease. It's because of the mutations in a gene called myosin 5B. So, for example, one can mimic now these in organoids and try to figure out how one can alleviate the symptoms of this disease. By using screening chemical libraries, one can also, if you have a mutant, cell line which is already existing, one can try to now cure this. And in fact, in fish, we do have a model which we ourselves have generated, which also can be used to test some of the genome editing related methodologies to cure, at least in animal models, can one get the wild type function back.
So the information from these model organism can be translated to some of the related fish models or animal models. That is something you see.
Absolutely.
Actually, we in plants also we have something similar. When we try to do some basic biology, we always do something on model organism like Arabidopsis. But the complexity increases when you try to do it into a kind of really non-model organism, even crops. So do you still see such challenges with genome editing from model to non-model organism?
Actually, I mean, it's a very good question. Some of the old gene editing methodologies face some of these issues, but CRISPR-Cas9 has become a very, very versatile system simply because of the ease with which you can work with it. So this particular platform can be applied and has been applied. In fact, it's being applied in variety of different organisms. And that's exactly the beauty of this, and that's exactly why it has been awarded the Nobel Prize so early, so quickly.
Agreed, agreed. Thank you, Dr. Mahindra. So we come to our next panelist, who is Dr. Mohan Murugesan. Mohan, welcome to this platform. We have been kind of listening a lot about Christian Medical College and your efforts about using base editing, prime editing for hemopoietic cell disorders. So can you expand more on it? क्या कर रहे हैं आप उधर, or how your research is going to help us on kind of getting some of the unsolved questions on gene therapies or something like that? Yeah.
So the current platforms are mainly based on like you have to go for lifelong blood transfusions. For example, if you take sickle cell disease or beta thalassemia, the patient like every month they have to go for the blood transfusions. So they get a lot of infections, and then like the pains or should be there, like it's a lot of problem. So what we want to develop is we want to take a patient's own cell. We want to take the blood forming cells from the patient, and then we also want to use the genome editing tool, because several of them have spoke about Cas9 base editor. All of them make a cut in the DNA, because if you want to raise particular part, we have to cut the DNA. But when you are cutting the DNA, it causes a lot of problems. So we have developed an approach where without cutting a DNA, we can correct the bases, like the alphabets in the DNA. That's the approach we want to follow. So the goal is like develop a one-time treatment approach. Instead of going like again back to clinic, we want to take a patient come one time to the clinic and take the blood forming cells, and then use this platform which more safer and then like very precise to correct the genetic changes, and then give it back to the patients, so then they can have a lifelong cure. So to prove that, we have started with beta thalassemia, because that's one of the common disorders we see it in the India, and then we also moving forward, we are planning to take it forward to sickle and then other immunodeficiency disorders.
Okay, so basically more on blood disorders you are trying to start with in your research.
Yes, the main advantage of blood disorder is, unlike other disorders, we can take out the blood cells outside.
Okay.
Because if I want to treat liver or brain, we have to act, as like Dr. Arka said, we have to give the genome editing tools inside the body. But
Okay.
for the blood disorders, we can take out the blood cells outside. We can do all the changes ex vivo, like in the plates, and then we can give it back to the patients, so that there is no harm, like there is no immune response, there is no other off targets happening in the body.
Okay, so that's come to a very interesting question again. What kind of scientific consideration and for these preclinical trials you need to be kind of taking care while doing all these studies? Because first, patient consent is definitely very important, but again, there are a lot of scientific and ethical considerations which comes into the picture. So can you expand more? What are the difficulties and challenges you are kind of dealing with on day-to-day for doing these kind of trials?
Yeah, so for the model wise, we first do it in the cell model, like cellular model.
Okay.
We do it because getting the patient sample is difficult. So first we check it everything in the patient sample. Like before we are going to that, we create a model in the cell and then prove things are working fine. Like we screen almost like 10 or 100 compounds, 100 guide RNAs, whether it's working on the cellular model. After we get the best platform, then we test it in the patient sample ex vivo, and then we'll test it in the animal model first, and then see whether after giving the blood cells to the mice, whether it's able to form the normal blood cells, and then whether it's able to cure the disease. We'll test it in the animal model, and then later we take it to the the phase 1 trial. That's the path it goes in. Like the problem what we face with taking this approach to clinic is like the cost, because as mentioned, like car therapy, like is like cost crores of rupee to go to the patient. So the approach what we are following is to bring down the cost to lower level. Like typically, if you want to do the therapy for these patients lifelong, it takes about like 50 lakhs. So we want to bring down the cost around 50 lakhs in one time. so that it can be, easily affordable to the patients. Even the 50 lakhs, some of them is covered by the government agencies, like, so that it can be afforded to all the patients in the near future.
I would request audience to have, if they have any questions, please do come up with questions. We can always take in between, not at the end, but if there are any questions from your side, please, I welcome them. Otherwise, I will continue with few of my more questions. Yeah. Yeah.
Hello, sir. Good afternoon to all. My question is that if we have used gene editing for curing some disease, so how would we manage the patient? Basically, after the editing, what are the monitoring for the patient? We have to monitor the patient for the long time, and how? What are the things that we look forward to it?
Arka, would you like to or?
Right. So that's a great question. So once we do editing, there are two risk items to monitor in any patient. One is the immune response, and the second is any kind of off-target editing that leads to formation of any malformations, hyperplasias, or even cancers. So these are the two things to be kept in mind. That's why gene therapy trials are very, very long. The initial response you are going to see in whatever 1 to 2 years, but all of these patients are supposed to be under long-term follow-up for up to 15 years to do that. So those are the two main.
Good afternoon, sir. Actually, I have question with the Bish, sir. In my PhD, I am also working with the secondary metabolites which give protection to the plants and work as the anti-nutritional factors. So is your lab is doing. So I am curious to know if we will reduce glucosinolates via CRISPR-Cas, doesn't it affect the plant health and yield?
So yeah, that's a very interesting question. And if you recall, what we are trying to do is that we are reducing glucosate only from the seeds. So we are not disturbing glucosate in any another plant parts. So that is something we are working in here is transporters.
Okay.
Some transporter which transport glucosinate from pod wall to seeds. So that only we blocked.
Okay, sir.
So now the plant will have only low amount of glucosinate in seed, which is good for health.
Okay,
But
sir.
the whole plant is going to have enough amount of glucosinolate for plant defense and immunity. So that's a kind of double kind of win-win situation for plant, that you have both nutritional factor as well as plant immunity at the same time.
Okay, thank you, sir. And sir, I'm also curious to know, as you mentioned, Sir's lab is editing Cas proteins, and we can use that. So I am curious to know that these Cas proteins are suitable for plants also, or only animals?
Oh, they're suitable for all living organisms, actually.
Yeah, so in fact, we are testing them and we are finding some very interesting result. And hopefully, we are working on some of the bottlenecks which we are finding are important for plant-based editing using those editors. And they are working very efficiently now. So we wanted to have it more so that it can be taken from lab to lab. So that is something going on. Yeah.
Good afternoon, sir. My question was actually a doubt regarding lifestyles. As I've heard so much time that if we take too much processed food or carbonated drinks, that does it cause cancer? And if it does cause, then how does gene editing help us?
Well, the short answer is yes, it can cause cancer. I think there's enough studies that are already showing an association. Theoretically, if you know that there is only one gene that is causing it, or a group of related genes that are causing that cancer because of you taking processed food of a certain kind, then you can introduce some sort of a mutation which is protective, just like he did in case of his editing, that he did only in the plant seed and not in the leaves. So you could potentially do that. But for that, it should be ideally one or very few genes that are associated with that progression. Thus far, what our research is telling us, that it's not a single gene, there are multiple genes that do get that. So it is a little more tricky to solve that.
Yeah, that's very nice. Yeah, please.
My name is Gaurav Govardhan. I have a question for Professor Sonawade. You talked about zebrafish model. I just wanted to know, I mean, maybe from all of you essentially, how do we decide on a particular model? I mean, which for a, I mean, I know that there could be a mouse model, there could be a monkey model, and now a zebrafish model or something else. How do we zero down on a choice of a particular model? Could you please elaborate that? Thank you.
Yeah, it basically boils down to the question that you have. I mean, if you want to study a very simple process like cell division, you can study it in a yeast, right? If you want to now look at the complex processes like development, there are other constraints that come into the picture. You of course can't use monkey because typically, I mean, if you look at the life itself, the time that is involved and reproduction and the number of progeny, all these factors become important. And typically when you study a process, you need to use approaches like genetics, molecular biology, and other cellular approaches. And some of these are also kind of sensitive to the number of progenies that you would get, right? And for zebrafish became actually useful because originally the developmental paradigms were established in Drosophila, which is it's a fruit fly. And turned out that eventually turned out that many of these genes are conserved also in humans. In fact, human development is kind of regulated by the genes which are identified in the fruit flies. But when this was not known, people thought that vertebrates like we would be different. Now, again, the studies cannot be done in humans, so people thought of using mouse or zebrafish as models. And zebrafish particularly because it gives more than 100 eggs at a time. So if you want to do genetics, it becomes easy. And also the embryos are transparent. So you actually can see through and you can look at the brain, you can look at the heart, you can look at the arteries, if blood is flowing fine, if your heart is pumping well. So you can actually identify defects much, much easily. And I think that was the advantage. And so there is no simple answer to your question. Again, it boils down to the question that you want to ask. And based on that, you will choose your model.
So can we say that both the zebrafish and Drosophila, they are equally good, but it's just that the embryo of Drosophila is transparent, so that is why it gives an added advantage, right?
Yeah, I mean Drosophila, zebrafish embryo is transparent, but there is a difference is that one is vertebrate, like it belongs to a kind of people, I mean us in some sense. Yeah. Which Drosophila is not. And initially the idea was that vertebrates, because they are more evolved, they will have some special genetic regulation, which turned out to be not the case in some sense.
So my question is, sir, one question with it for DMD, sir. So one of my relatives is having DMD in their genes. So mother, we know that is a carrier. So I have two questions. One is that for therapy purposes, what is the right age to come for therapy if we know that the child is already suffering from DMD? So if we want to have a therapy, we know that they have limited age span. So can we, the sooner the better, or there is a capping that after this age nothing can be done?
Sooner the better. Even before the disease manifests is the right time actually to get a therapy which will last through the lifetime. Once the disease has set in, so the disease is happening because of mutation in dystrophin, but because the disease has happened, the muscle cells are dying. So there's a lot of inflammation, necrosis, fibrosis, etc. that is happening, which is secondary to the primary pathology. Now all of these become barriers to the therapy. So now once they become immobile and wheelchair bound, at that point there is significant amount of inflammation and immune activity and fibrosis, particularly in the heart and diaphragm. So, and there is a lot of muscle loss. So at that point, even if I correct or put the correct copy of dystrophin there isn't enough amount of muscle tissue remaining to generate enough force for you to reverse the condition. And there is these barriers of fibrotic tissue and immune activity which prevent the disease from working. So this is data that we have from both human trials and animal models, that if you give the same dose of the drug to a healthy subject and a late stage disease subject, the drug works maybe at a 30% efficiency, because the immune cells, they get rid of all of the cells that get transduced by the drug. So that is a major barrier. So for any disease, prophylaxis or very early intervention is the best way to ensure that you have a long lasting effect. Now that's not to say that later on it can't work. It can work, but the complexities of the pathology make it more difficult.
So the other part of this question is like, since mother is the carrier, so are you focusing on that direction also? Something can be done with respect to that X chromosome, like because the gene is there and at the carrier level rather than at the.
That is a question relevant to all rare diseases and genetic diseases, including cancers. But as of now, germline DNA modification is illegal in the country. So to correct it in the mother, you would have to do germline modifications. So that is generally illegal everywhere.
Thank you, ma'am.
I would like to ask, if Dr. Mohan would like to add on something on this part, because all CRISPR-based trials, what are the regulatory bottlenecks you see right now, and what is the way forward for Indian ecosystem? What do you think should be done for these CRISPR-based trials?
For the CRISPR-based trials, mainly the bottleneck we face is like the regulatory approvals to get a scale-up data. Because generally, if you want to scale up from the proof of concept to the larger scale, we see a lot of GMP regulatory challenges, like mainly like importing and getting approval. And those are the things, if it is fine-tuned, it may go to the easy path. And also like each component, when it goes for the CDSCO approval and to the next stage, it takes longer period of time. So now the government has introduced two-way policy that also going to make it faster.
So is there any guidelines or framework we are following in Indian subcontinent, in India, in our country, to follow these GMP practices and also for CRISPR-based trials? Is it so?
Yes. CDSCO has a fine regulatory approval for each product to go to the particular stage.
Mm-hmm.
So they have a guidelines to follow from the GMP level to the phase 1.
And compared to countries, US or something, how fast or how lethargic our ecosystem is getting these approvals?
The approvals now, like before, it was to take time, but now things are getting faster. So during the the coming years, it will get faster.
And I would like to listen from Dr. Ghosh also, because about your experience on getting these approvals, how definitely these were very challenging, but now how the scenario is changing over the years. Please comment on that.
So when we started working on it, on gene therapy in India, there were no guidelines for doing gene and cell therapy clinical trials in India, right? So the guidelines came in 2019. So we were all co-authors on the guidelines. And from then on, things have sort of streamlined to an extent. But that was the first version of the guidelines. We were supposed to have revised the guidelines several times by now, but that all hasn't yet happened. Now there is a fresh guideline that is going to come from CDSCO. It should come anytime now. So previously, the problem that is there with the regulatory was that biological or living drugs are completely different from chemical entities. Right? Chemical entities like, say, ibuprofen or something is just one molecule. Now, when you're talking about DNA, you have a billion molecules, right? So all of them have to be arranged in a certain way. They have to be produced in a certain way. So you have to use biological factories to produce it and not a chemical factory with a defined instrument. So that is the trouble. So therefore, the regulatory also has to check so many different things. And some of these checks that they know from the pharma world don't apply to the biologics world. So that gap in approach is always there, which causes some amount of delay. But now I must say that everything has gotten faster. It should get even faster, but we are getting there, I feel.
Yeah, and I'm sure that cost will also come down. And I was talking to Dr. Arka also, that possibly in few years we will see these, even the insurance people might also kind of if it will be good if we can get all these within insurance, then the kind of the bearing of all these therapies will be kind of very public oriented. Yeah, and I look forward to see India to be going in that direction in next 4 to 5 years. Yeah. Yeah.
Yeah, one last quick question, please.
Sir, I have a question regarding CRISPR-Cas9. So, sir, CRISPR has been integrated to detection system also. So there has been development of CRISPR-Cas9 based detection for pathogens. So my question is that what is the best approach if we want to go into point of care diagnostic using it?
So I think Devo's lab developed the point of care diagnostic for both sickle cell as well as for COVID.
Yes.
So he used a paper-based method where, you know,
So, like, sir, they have been using CRISPR-Cas9 in that direction.
Right.
What are the other tools that we can use? Like, recently there has been TNP-based gene editing tool for India. So, can we go in that direction with you, with the help of that tool also?
Yeah, it will all.
I've been very fascinated about that discovery.
Definitely, it can come through, and it all depends on the preciseness of your editor. If your editor is very precise, it will definitely able to kind of differentiate between even each and every pathotypes. So that is something, yes, in future it can be done.
Yes.
And we have good example from Devo's lab. We have where we are able to do at least for few diseases like even COVID and sickle cell anemia. And I'm sure for many cancer, different type of cancer, this can also be something can be done in near future. So. Yeah.
Thank you so much, sir. Thank you.
Yeah, thank you all for being such a patient audience. Now, I extend my sincere gratitude to all the panelists being here on the stage. Now, may I request to Dr. Rajesh sir to present mementos to our esteemed panelists. Yeah, to Dr. Arksubra Ghosh sir. Dr. Mahendra Sonamne sir. Dr. Mohan Kumar sir. Dr. Naveen Chand Bish sir. So with this, I thank all the people present here. So now, okay.